All right, so let's go back. I think my recording was cut short on the first time, so I'm having some technical issues. But let's proceed with your platelets again. So again, your platelets are enucleated cells. Okay, are your platelets are enucleated cells. So meaning to say they don't have nucleus. So for this very reason, in some references, they don't actually consider your platelet as a true cell because it doesn't have your plate. They doesn't have nucleus in it. Your platelet by size is actually just 2.5 micrometer. So imagine that, no? Imagine coming from a 30 to 50 micrometer megakaryocyte. Now you're just 2.5 micrometer. So it's very important for you guys to also remember that. In addition to that, okay, in addition to that, our your platelets, okay, your platelets, with regards to their count or their quantitative count, it is around 150 to 400 times 10 to the ninth power times per liter. Okay, so 100 to 400, 150 to 400. Although consider they are considerably higher in women, so higher in women, but when men and women reaches the age of 65, it is slightly lower or on both sexes. Okay, so higher in women, but when men and women reach the age 65, their platelet count would tend to be lower. Okay, would tend to be slightly lower. So your platelet has two population. So similarly to your neutrophils, correct? You have the marginating and the circulating neutrophils. Your platelets has also two population. The first one comprising 30% of the total the total number of your platelets, or one third of the total platelets are seen in your spleen. Okay, are seen in your spleen, and we call them collectively as your resting platelets. Okay, we call them your resting platelets. On the other hand, we also have the two-thirds or the 70% of the remaining platelets which are circulating in your peripheral blood. That's why we call them collectively as your circulating platelet. So, inside the laboratory, medical technologists can perform qualitative count for your platelets. And how do we assess qualitatively the number or the the number of platelets, whether it is adequate or inadequate? That is by counting 10. Okay, 10 oil immersion fields. So we are counting at 10 oil immersion fields, and normally, okay, if your platelets are adequate, you would actually see around 7 to 21 platelets per oil immersion field. Again, you will be seeing 7 to 21 oil immersion field per field of on your oil immersion field. Okay, so 7 to 21 platelets. Okay, so please take note of that. So if we have 7 to 21, so take for example, you had a total of um you were able to count um ranges within 7 to 21 and all the 10 oil immersion field, then we can say that the platelets are adequate. Okay, the platelets are adequate. So moving forward, we also have your of course, please also remember that your platelets, okay, your platelets actually vary in size and in your shape. Okay, they vary in size and they vary in shape. That but normally, I don't want everybody to take note of this. Your circulating and your resting platelets are actually biconcave. Okay? Meaning to say, the platelets within our body are actually biconvex. Okay? Biconvex in shape. But upon collecting blood on your purple top, or your containing your K2 EDTA, okay, your K2 EDTA, your platelet tends to round up. Okay, your platelet tends to round up. Please remember that because baka some of you would be confused with the shape of your platelet. Within, okay, if we're talking about the circulating and the resting platelet, they're actually biconcave in shape, biconvex in shape. But if your platelets now are collected on your um, if your platelets now are collected on your EDTA tube, they tend to round up. Okay, they do tend to round up. So very important terms to remember with regards to your platelets are your chromomere and your hyalomere. Your chromomere, okay, your chromomere are the granular part, which are located at the center of your platelets. Okay, these are the granular portion of your the granular portion of your platelet. Let me show you a picture of a bigger platelet. So, ayan, I'm advancing, but yeah, this is your chromomere. And the one that surrounds the chromomere, the non-granular, the clear light blue, we call that your hyalomere. Okay? We call that your hyalomere. So, take note of that, guys, ha? The one that you are seeing, okay, the one that you are seeing with granular and are centrally located, we call that your chromomere. On the other hand, the one that surrounds it that are colored clear to light blue, okay, the one that surrounds it are your hyalomere, okay, that those are your hyalomere. So normally, this is how your platelet would look like. So nakikita, medyo maliit, no? But that is already the oil immersion field. So the one that you can see, the purplish center, that is actually your chromomere. And then the extension of it, the one that surrounds your chromomere is already your hyalomere. Okay? So please take note of that. Okay, your chromomere and your hyalomere. So moving forward, let's talk about the distribution and the kinetics of your platelets. So the maturation time of your platelet is around 5 days. Okay, so it would take 5 days for your megakaryocyte to mature within your bone marrow. So the newly released platelets are usually okay. The newly released platelets are usually larger. They are also more metabolically active compared to other platelets, and they are more effective hemostatically. So when I say effective hemostatically, remember the first one that we discussed about um, your primary, your secondary hemostasis. So, 'di ba? It's just normal. The younger the cell, the more active it is, the more effective it is. Okay, the more effective it is. So that is also the same case with your platelets. The newly secreted or the newly released platelets, or when I say secreted, released, sequestered, all are the same terms. Okay, so the newly released platelets are actually larger. They're also more metabolically active, and they are more effective hemostatically. So please remember that. Please remember this characteristic of a newly released platelets. And of course, um, your platelets, once released, they can survive in your circulation for around 8 to 9 days only. Okay, they can only um, they can only leave on your in your blood vessels for 8 to 9 days. Okay, for 8 to 9 days. So these are the characteristic of a newly released platelets. Now let's go to another type of platelets. Platelets, okay, which are your reticulated platelets, in a sense that they still have residual RNA on their cytoplasm or on their hyalomere. Okay, on their hyalomere. So these stress platelets carries free ribosomes and fragments of rough endoplasmic reticulum. So as you can see, meron pang RNA, meron pang ribosomes and rough ER. Okay? These stress platelets are considerably bigger compared to the normal platelets, because your normal platelets would just average to 2.5 micrometer, and your stress platelets would average to 6 micrometer. That is the normal. But during the release of your stress platelet, we have around 12 to 14 femtoliter. Okay, 12 to 14 femtoliter and 6 micrometer in size. These stress platelet also tend to round up. Okay, they also tend to round up if collected in your EDTA tubes. But if collected in your citrate, they actually appear to be cylindrical and beaded in appearance. Okay, cylindrical and beaded in appearance. So it's very important. Okay, it's very important to identify these stress platelets because the release of stress platelet is just a response of your body in cases of extreme thrombocytopenia. So makikisulat po, your stress platelets are released in your peripheral cell, in your peripheral blood in cases of extreme thrombocytopenia or extreme cases where your thrombocytes are really low. Okay? Really low. So your stress platelets can be identified using your flow cytometry, your flow cytometer. So the first thing that you would do is to incubate the whole blood with your nucleic acid dye, which is your thiazole orange, and it would bind now to the RNA that are found on your endoplasmic reticulum. Okay, remember that you're in your endoplasmic reticulum, correct? Your endoplasmic reticulum is seen in your stress platelet. So how would you do that? Um, how can we um, how can we identify that there are stress platelet using flow cytometer? There would be a positive fluorescence. So a positive result is actually a 3 times, okay? 3,000 times increase when it comes to the fluorescence, okay? 3,000 times increase when it comes to their fluorescence, okay? How does your stress platelets looks like, okay? Your stress platelets looks like this, okay? Your the one here, okay, this is your stress platelets, considerably very bigger, so 6 micrometer, very much close to your 6 to 8 micrometer RBC already. So this one, the one that I am pointing on your screen right now, the small dots, those are actually your normal platelets. Your normal platelets that actually are within the size of 2.5 micrometer only. Okay? 2.5 micrometer only. So having said that now, okay, having said that now, please do remember that your stress platelet are not normal. Okay? Your stress platelets are not normal. Your stress platelets only appear in your peripheral blood when there's a case of extreme thrombocytopenia. Okay? So again, moving forward, huwag kakalimutan, chromomere, hyalomere. Do not forget the maturation day within the bone marrow and the lifespan of your platelets, and also the normal value, the normal sizes, the percentages of which being in the circulating pool and those that are in the resting pool. Okay, now let's go to the ultrastructure of your platelets. So these are your platelets. If you're gonna look at your microscope, okay, these are your RBC. You see the central pallor. This is your neutrophil, a polymorphonuclear leukocytes. You can see it's multi-lobe. It's actually a segmenter. Okay, this tiny fragments here are your platelets. Okay, those are your platelets. So now let's talk about the normal platelet ultrastructure. So even if your platelets are very tiny and small, they actually have different parts within them. So in the ultrastructure of your platelets, you can see their microtubules. They also have mitochondria, their dense bodies. They also have lysosome. They have a surface connected to the canalicular system. We have your peroxisome. They also have glycogen, where they store up their energy. They have their plasma membrane, and of course, they have their alpha granules and their dense bodies, which are very important as we go along. Okay, so first let's talk about the glycocalyx. So the glycocalyx is the outer membrane surface of your platelet. Your glycocalyx are usually 2 to 30 nanometer in thickness. Okay, so what is the main characteristic of your glycocalyx? They are adhesive. So when we say adhesive, they actually attach themselves. Okay, they actually attach themselves to your blood vessel. That is the reason why, okay, that is the reason why during, isn't it, during breakage or during injury or damage in your blood vessel, your platelet would act, the first thing that they would do is to adhere, okay? Upon activation, they would adhere in the blood vessels, okay? Once that your platelets are activated, they will now start to adhere on your blood vessels or wherever the site of injury is. They maintains the negative surface charge of your platelets, okay? They maintain the negative what do you call this, the negative surface charge of your um platelets. So they also absorbs albumin, they absorbs fibrinogen and transports them to store them on the organelle through endocytosis. Okay, so through endocytosis, your platelets actually absorb albumin and fibrinogen to store them up within the platelets. Okay, to store them up within the platelet. Next, after your glycocalyx, okay, after your thick glycocalyx, okay, after this thick glycocalyx here comes now your plasma membrane. Your plasma membrane, similarly to all other cells, is a semi-permeable membrane. They are selectively permeable. So we have cholesterol, fatty acids, and carbohydrates here. Of course, every structure of your as you all know, every structure naman of our cell, okay, is comprised of a lipid bilayer. So you have your cholesterol here that stabilizes the membrane and also maintains the fluidity, okay, the fluidity of the plasma membrane. Your cholesterol also act as a control. It also controls the passage of material within your cell, so allowing only specific cells to enter within your plasma membrane. So you also have their tails of fatty acids, that comprise your lipid bilayer, and of course you have your carbohydrate side chain that would extend into the plasma to form your receptors. As we go along, okay, as we go along your platelets, we would be discussing there the GP2B, the glycoprotein 2B, that are actually receptors within the platelets. Okay, those receptors of your platelets are very important, specifically when we discuss the different types of diseases and disorders or abnormalities with regards to your platelets. Okay, so I hope I made myself clear with your plasma membrane. Okay, of course, if you have your cholesterol, your fatty acid, and your carbohydrate, do not forget, of course, your phospholipid bilayer. So you have your phosphatidylcholine, your sphingomyelin that are in the plasma layer. Of course, always remember the most famous, your phosphatidylinositol that supply your arachidonic acid, your phosphatidylethanolamine, and your also phosphatidylserine that flips and where coagulation enzymes assemble. I want you to remember this. Your phosphatidylinositol is the one that supplies arachidonic acid in your platelets. Arachidonic acid, I want you guys to keep that in your heart, in your mind, because we will be talking about our arachidonic acid shortly. In addition to that, your phosphatidylserine, okay, your phosphatidylserine is where um your coagulation enzymes assemble, okay, during, of course, obviously, during coagulation. So those are two very important phospholipid um phospholipid phospholipid different those these are very important kinds of phospholipids, okay, phospholipids that are found within your cell, okay, not within but on your plasma membrane rather. So we also have here your dense tubular system. Your dense tubular system are derived from your rough endoplasmic reticulum. So they have a positive staining for platelet peroxidase activity. So meaning to say, um the peroxidase, they contain peroxidase, okay. So when staining platelets, to check their peroxidase activity, the positive results of your platelets are due to your dense tubular system. Your dense tubular system is also the site of arachidonic metabolism within the platelets. Okay, within the platelet. So the site of arachidonic metabolism again is your dense tubular system. In addition to that, your dense tubular system also provide a low level of cytoplasmic calcium in the resting platelet. So they also act as a calcium sequestering pump. Okay, so your dense tubular system acts as your calcium sequestering pump. Ladies and gentlemen, they are colored such not just for design but for importance. So again, your dense tubular system is the site for your arachidonic acid metabolism and your calcium sequestering pump. Sir, para saan po ba? As we go along, this prelims, we'll be talking about your vascular intima, your primary and your secondary, your primary hemostasis and your secondary hemostasis. You will be meeting arachidonic acid and your calcium sequestering pumps very, very soon. So, having said that now, okay, your dense tubular system also controls your is actually the control center of your platelet activation. So remember, during platelet activation, they activate, they they secrete and they would aggregate. So your dense tubular system is actually the control center for platelet activation. It bears a series of enzyme. Okay, it has a series of enzymes that support platelet activation. And what are those enzymes? Those enzymes that would actually promote platelet activation are your phospholipase A2, your cyclooxygenase, your thromboxane synthase, which are very important for prostaglandin synthesis, and also your phospholipase C. Okay, that supports the production of your inositol triphosphate or your IP3 and your diacylglycerol or your DAG. Okay? So again please remember that your dense tubular system, 'di ba? Ang amazing ng platelet, napakaliit, very tiny when you look at it under the microscope, but it is far more complicated. That's why it is very separated into your hematology too. So again, your dense tubular system, okay, your dense tubular system contains the following enzyme. Please remember the enzymes and please remember what specific function does this enzyme does within your body. So moving forward, we also have your surface connected canalicular system. Your surface connected canalicular system is the direct communication of your platelet in the extracellular environment. Okay, in the extracellular environment. So remember that your platelet no longer have your it no longer have your nucleus. Okay, that's why it needs other alternatives into communicating into the external or the extracellular environment. So in addition to that, the SCCS is also the route for endocytosis and secretion of granular contents on activation. So once that your most of the activators, okay, most of the activators that will be coming extracellularly, okay, yung mga mag-activate doon sa platelets will be undergoing endocytosis and um, will follow the route of your surface connected canalicular system. At the same time, when it comes to exocytosis or the secretion now, exocytosis, the release or the disposal or the secretion of granular products, okay? The secretion of your granular contents upon activation are also happening on the SCCS, okay? Or the surface connected canalicular system. A question, where does the calcium sequestering pump is located? Where does the calcium sequestering pump is located? Okay, that is your dense tubular system. Okay, aside from that, um, this is also the route of the secretion of your alpha granule content. Remember, 'di ba? Sabi ko nga kanina, your platelets after being activated, the next step will be platelet secretion. What will it secrete? It will secrete their granular content. In addition to that, they will secrete their alpha granule content. And how does your platelet secrete those materials? The answer is through the surface connected canalicular system. Okay, now let's go to the structure of your now let's go to the structure into 25 nanometer in size. So it make up the mitotic spindle fiber that are seen in your platelet. They are very important because they maintain your platelet shape. Okay, they maintain your platelet shape. They are also called your circumferential microtubules. Why circumferential microtubules? So let me show it to you because they are actually surrounding your platelet. So as you can see, these are your microtubules. Okay, these are your microtubules. We call them your circumferential microtubules. Okay, because they maintain the shape of your platelet. In addition to that, they're also composed of your tubulin. Okay, your tubulin, which are the structural support for your normally discoid cell. So these are very much evident within your body, specifically your circulating and your resting platelets, which are biconvex in shape. Okay, biconvex in shape. We're nearing the end of our discussion, so please hold on. So we also have here um, a very important function of your microtubules because they also are the one that contracts. So once your platelets are activated, they contract. Okay, and when they contract, they encourages now the expression of your alpha granules. And when they contract, as they express, okay, as they encourage the expression or the secretion of your alpha granules, saan mapupunta si alpha granules ulit? Where would this alpha granules be secreted? Correct. That is through your surface connected canalicular system. So after your cytoskeleton, okay, very important thing to remember are also your microfilament. Your microfilament are composed of platelet actin and myosin that interacts to form your actomyosin. Collectively, we call your actomyosin as your thrombus teni. Okay? We call them your thrombostenin. Your thrombostenin is very important for clot retraction. Okay? Makikitanda, for clot retraction, we have your thrombostenin or your actomyosin. Actomyosin or your thrombostenin for clot retraction. The things that you are hearing me say, platelet secretion, clot retraction, just store them up. Write them down if you want to. Okay, write them down. Those unfamiliar terms, because when we go to the succeeding topics, you'll be able to meet them and understand what are their purpose for. So of course, your actin, ayan, your actin causes your platelet to contract, okay? Your actin causes your actin, okay? Your actin causes your platelet to contract. In addition to that, it also anchors their plasma membrane glycoproteins and proteoglycans. So remember, on the surface of your plasma membrane, you have your carbohydrates there that serves as your antigens. Those are your glycoproteins. They also present they are also present throughout the cytoplasm, constituting 20 to 30% of the platelet protein. So please take note, okay, that in resting platelet, your cytoplasm is only at its globular and amorphous form. Okay, as the cytoplasmic calcium concentration increases, okay, as your calcium concentration increases, your actin become more filamentous and they are now contractile. Okay, because they are now ready to be activated. Okay, they are now ready to be activated. So, makikitanda ako, no? Maybe you're wondering, bakit ang daming calcium? Okay? But ang daming calcium because calcium is a very important cation when it comes to your platelets. Okay? It is responsible in their activation. It is responsible in the formation of a filamentous and contractile actin. It is also responsible for the coagulation. That is the reason why, di ba, if you can remember, most of your anticoagulant, your EDTA, correct? Your citrate, they are all, what are their action? It is all for the active, it is all to, um, to chelate or to bind to calcium. That is to prevent your platelets from being activated, therefore causing a coagulation in your blood. Okay? So, hopefully, nare-realize nyo na bakit kaya bind to calcium, chelates calcium. Okay? That is the reason why. Okay? So, let's go now to the different granules of your platelet. First thing are your alpha granules. So, your alpha granules can be seen on your platelets. Okay, so they average around 50 to 80 alpha granules per platelets. Okay, your alpha granules are very important content of your platelets because they participate in the adhesion, aggregation, and plasma coagulation. Okay, they participate in the adhesion, aggregation, and plasma coagulation. So remember, like I told you, what I told you a while back, that your platelets would adhere to the site of injury. Not only is component of the platelet is that, okay, correct, your glycocalyx. Aside from your glycocalyx, okay, your alpha granules also participates in the adhesion, but not only in the adhesion but also in the aggregation and the eventual coagulation within your plasma. So your alpha granules would stain gray in osmium dye. Okay, so under transmission electron microscopy, okay, you would see your alpha granules colored gray. Okay, that is the reason why. Okay, that is the reason why we actually have a type of platelet syndrome. We call that gray platelet syndrome, which is inherited because of the absence of your alpha granule content. Siguro magtataka kayo, akala ko sir kasi masyadong madaming alpha granules. No, your gray platelet syndrome, okay, is not the abnormal abundance of your alpha granules, rather the absence of your alpha granules. Okay? So please remember that. Okay? Your alpha granules, again, participate in your adhesion, aggregation, and coagulation. It is colored gray when dyed with your osmium, seen under your transmission electron microscope, and they are very much common in your gray platelet syndrome, not due to the absence of your alpha granules. What does your alpha granules contain? Okay, what are the content of your alpha granules? Your alpha granules contains your fibrinogen. Remember remember that your glycocalyx, okay? Remember that your glycocalyx absorb albumin and your fibrinogen. So your fibrinogen is being stored up on your alpha granules. They also have your VWF. We're not talking about Wonder Woman kahit mukhang Wonder Woman yung sign. But we are talking about your von Willebrand factor. It is your von Willebrand factor. Aside from that, your alpha granules also contain your factor 5 and factor 8. Okay, factor 5 and factor 8. So very important to remember that. In addition to that, your alpha granules also contain your platelet factor 4. What is your platelet factor 4? It is a it actually is a a a substance. Okay, your platelet factor 4 is a substance that would neutralize your heparin. Okay, it would neutralize your heparin. Why? Bakit heparin, sir? Because your heparin, isn't it, is the natural anticoagulant within your body. Okay, so it regulates, in addition to that, your platelet factor 4 also regulate vascular permeability. Okay, vascular permeability, and it also regulate calcium mobilization from your bone. Why is calcium mobilization from your bone important? Again, let's go back to your calcium sequestering pump in your dense tubular system and the action of your calcium, more specifically during coagulation. Okay, so it also act as a positive chemotaxin. Okay, your platelet factor 4. I hope you're finished, isn't it? In your immunology, you also discussed diapedesis chemotaxis. So your platelet factor 4 acts as a chemotaxin for your monocytes and your neutrophils. Okay. In addition to that, what else can we see on your alpha granules? Aside from this, we also have your P-selectin, okay? You have your P-selectin, your beta thromboglobulin or your BTG, and we also have your platelet-derived growth factor or your PDGF, which repairs now your damaged blood vessel. Okay, 'di ba? It's a eureka moment. Okay? Remember, sabi natin last time when we discussed homeostasis in the fibrinolysis, your platelet would remain on your blood vessel to not only stop the clot but also eventually to repair the clot, to repair to repair the to repair to repair our blood vessels. Okay, so that is the action of your platelet derived growth factor. So in addition to that, aside from alpha granules, we also have your dense granules. So your dense granules is just few. Okay, you only have 2 to 7 dense granules per platelet. So they are characterized to be bullseye. Okay, they are dense bodies and they stain black using osmium dye. Okay, so if your alpha is gray, your dense granules are black when stained with osmium. So the dense granules is actually a location or a locus of storing your non-metabolic pools. Okay, your non-metabolic substances like your ADP. Okay, that would support your aggregation, your ATP that is being used to detect that is being used in the detection of your lumi aggregometry. Okay, in your lumi aggregometry. So your ATP is the one that is being detected. So that is your luciferase luminescence. So this is a type of a this is a type of a test that we do. This is a type of test that we do to check the ability of your platelets to aggregate. Okay? 'Di ba siyempre alam natin merong yung platelets nag-a-adhere. Yung platelets mo ma-activate, mag-a-adhere, magse-secrete ng substances through the dense tubular system ay due to through the SCCS, okay? And then it would also aggregate. So, paano natin malalaman that it is capable of aggregation? We do your Lumi aggregometry, okay? Or your luciferase luminescence. So, your serotonin also is found on your platelets, okay? Specifically in your dense granule, okay? Which makes sense now, okay? Isn't it that one of the primary response of your body during trauma or damage, anong gagawin ng katawan natin? Vasoconstriction. But sometimes that would not be enough. That is the reason why, okay, nakakuha niyo yung beauty, yung collagen na na-release sa blood vessel, okay, in the coming days, I'll give you on our synchronous class, we'll discuss this on Monday. So, ina-imagine niyo 'no, nung nasira yung blood vessel, na-secrete si collagen. Si collagen mo ngayon, i-a-activate niya si platelet. At si platelet ngayon, maglalabas din siya ng serotonin to even enhance your vasoconstriction. So your serotonin is a vasoconstrictor that would promote the constriction of your blood vessel to prevent further blood loss. Okay? Of course, your dense granules also contain your calcium and your magnesium that are very much important in the activation and the coagulation. Okay? And of course, do not also forget that these dense granules would migrate to the plasma membrane and release their content directly into the plasma on activation. Sir, ganun po yun? Yes. As you remember, di ba, when we discuss your surface connected canalicular system, we are only talking about this is the route of secretion for alpha granules. Okay? Your SCCS are only for your alpha granules. Si dense granules, maagap. Kasi it contains your serotonin, it contains your calcium, and it contains substances that are very important for your platelet aggregation and even your blood vessel constriction or your vasoconstriction. So your dense granules, um, they won't go to the surface connected canalicular system anymore, but rather they would directly go to the plasma membrane and release their contents there directly to the plasma. Okay, directly to the plasma for upon activation. So, in addition to that, we're nearing the end. So, your platelet granules, okay, ladies and gentlemen, um, also contain your lysosomes rather. Your lysosomes would stain positive with aryl sulfatase, with B-glucuronidase, your acid phosphatase, and your catalase. So, this is the different stain that these are the different components of your lysosomes. Okay, so their main goal or main objective within the cell is to digest the vessel wall matrix component during in vivo aggregation. Okay, so they digest vessel wall matrix component during in vivo aggregation. So remember that they digest um, vessel wall matrix component during in vivo aggregation. Okay, and of course they also digest autophagic debris. So take for example, may na-take up yung platelets mo na substances, they would actually digest those debris within your cell. Okay? So in summary, so I want you guys to remember this summary that I will be having. So in the peripheral zone, okay, we have four zones kasi. We have four zones of the platelet ultrastructure. What are those four zones? The four zones are your peripheral zone, your soul gel zone, your organelle zone, and your membranous zone. What are the components of your peripheral zone? Your glycocalyx and your plasma membrane. Your soul gel zone are your microtubules and your microfilaments. Your organelle zone is where your alpha granules, your dense core granules, your lysosomes are seen. And in the membranous, we have your dense tubular system and your surface connected canalicular system. So please remember and memorize not only the different function of these ultrastructures, but also onto which zone do they belong. So if you have any questions or clarifications, I will be putting up our discussion board on our TLC. So that is it for today. Thank you so much for listening. And if you don't have any questions or clarifications, you can just simply go through. You can just simply go through our post activity for today and finish that until Tuesday. So again, thank you so much for listening and have a great day.